Hello, everyone, and welcome to the Pediatric Dermatology Research Alliance webinar on exploring the future of TYK2, cutting-edge research and therapeutic implications for pediatric dermatology. I am Jen Dawson, PeDRA's Associate Director of Educational Programs, and I am absolutely delighted to introduce your moderator for this session, Dr. Kelly Cordoro. She is a professor of dermatology and pediatrics and the McClelmont Family Endowed Professor of Pediatric Dermatology at the University of California, San Francisco. She is Division Chief and Fellowship Director of Pediatric Dermatology and a member of the Academy of Medical Educators at UCSF. Dr. Cordoro cares for children with all skin conditions and has a special clinical and research focus on complex inflammatory and autoimmune diseases, especially psoriasis. Early in her career, Dr. Cordoro received a Competitive Career Development Award from the Dermatology Foundation to study pediatric psoriasis and subsequently became a founding member of the Pediatric Dermatology Research Alliance and the inaugural co-chair of PeDRA's Psoriasis Investigator Group. She has served on the medical board for the National Psoriasis Foundation and is active in the International Psoriasis Council. Dr. Cordoro co-founded and co-directs the Pediatric Rheumatology Dermatology Multidisciplinary Clinic at UCSF, and she was the lead author on the first ever joint AAD-NPF management guidelines for pediatric psoriasis, published in 2020. Her clinical, scholarly, and educational contributions have been consistent and diverse, and I am thrilled that she so enthusiastically accepted our request to moderate the webinar today. So, Dr. Cordoro, I am going to turn it over to you to introduce our speaker.
Fantastic. Jen, thank you so much. And PeDRA, my beloved organization that I've been involved with from the very beginning, I'm incredibly delighted to have such an esteemed guest and webinar this morning. I have the distinct honor of introducing Dr. Gudjonsson. Before I do, I'll state that I have no relationships with industry or any conflicts of interest to disclose. Dr. Gudjonsson is with us this morning from University of Michigan. He is an MD, PhD, and the Arthur C. Curtis Professor of Skin Molecular Immunology and Professor of Dermatology, Internal Medicine Division of Rheumatology, and a Taubman Medical Research Institute scholar at the University of Michigan. Dr. Gudjonsson's primary research focus is basic immunological and genetic research on chronic inflammatory skin disease. This is a match made in heaven for he and I to be doing this together. He has published over 320 peer-reviewed papers in top-tier journals and has won numerous research awards, including awards from the American Skin Association and Doris Duke Foundation, and has multiple NIH grants. Dr. Gudjonsson was selected to present the Society for Investigative Dermatology Rising Star Lecture in 2018 and has gone on and continued his outstanding and impactful work. He was elected as a member of the American Society for Clinical Investigation in 2020 and the American Association of Physicians in 2024. He's currently a fellow of the American Association for the Advancement of Science, and he is the director of an NIH-sponsored P30 Research Core Center at the University of Michigan. He serves on the Society for Investigative Dermatology Board of Directors and is a scientific counselor to NIAMS. NIAMS is the National Institute of Arthritis and Musculoskeletal and Skin Diseases. I feel incredibly grateful to welcome you, Dr. Gudjonsson, to provide your most important remarks about this really interesting topic, and then have time for discussion after your formal comments. So without further ado.
Yeah, thank you so much, Kelly, for that very kind introduction. I'm very excited to be with you today, and I'm going to talk to you about a topic that I've actually been looking at for quite a while, both genetically and immunologically. Just as a background, a lot of my time is spent on research, but I also do quite a bit of clinic, and I run, now it's only half a day a week of clinic, but I manage mostly inflammatory diseases, both in adults and children. So I'm very excited to be with you today and show you a little bit of how we kind of look at these things both from the clinical and the molecular kind of a perspective. So the title of my talk is going to be on exploring the future of TYK2, cutting-edge research and therapeutic implications for pediatric dermatology. These are my disclosures over here, and just as a brief introduction about TYK2 signaling. So TYK2 was actually the first member of the JAK family that was described. And this is under the family of what we call like cytoplasmic tyrosine kinases. So I have a figure here on the right that kind of shows you the whole tyrosine kinase family. It's multiple different members that have very variable and different biological function. But you have here in the red, you have the receptor tyrosine kinases, and then you have in the blue the cytoplasmic tyrosine kinases. And what I have here is to kind of show you the grouping of these four members, the JAK family, which TYK2 was as I said, the first member. So TYK2 is a tyrosine kinase. It's an enzyme that can transfer a phosphate group from a molecule called ATP to tyrosine residues or specific proteins inside a cell. And this shows you, this is a cytokine receptor over here, and you can see this very complex machinery that is operating on the cytoplasmic side of the cell and then transducing the cytokine signal from outside the cell into the nucleus of the cell through these STAT proteins and activating a transcriptional regulator of inflammatory responses. So TYK2 really has a key role in functioning as an on or off switch of multiple different inflammatory signals that I'll go into in details in a little bit of a time. So in terms of the objective of my presentations, right, I want to provide an overview of TYK2 and the JAK family and its role in the immune system. I also want to give you a brief overview of immune-mediated diseases where TYK2 has been implicated in their pathogenesis, and also kind of touch upon the two main classes of TYK2 inhibitors that we use in the clinic. The ones targeting the active domain or the kinase domain of TYK2, we call these kinase inhibitors. And then the ones that target the regulatory domain or the pseudokinase domain of TYK2, and we call those allosteric inhibitors. And then I'm going to briefly touch upon some of the ongoing clinical trials, including pediatrics, where TYK2 inhibitors are being used. To move forward and kind of go back a little bit towards the JAK-STAT signaling. So there are four family members within the JAK family: JAK1, JAK2, JAK3, and TYK2. This shows you the structure of those proteins. It's the same across all four members. It has these different domains that have different function in terms of how they bind to proteins inside the cell and how they mediate signaling. But the key ones are the pseudokinase and the kinase domain of these receptors. The JAK family members mediate downstream signaling through the STAT family of proteins, and that has multiple different members that then kind of link back with the JAKs and the TYKs here underneath the cytokine receptor, and through dimerization kind of go to the nucleus and activate a different inflammatory program depending on which STAT actually is being used. And these also have a very conserved and similar structure in terms of how they work, including a DNA binding domain to kind of mediate activation in the nucleus. And if you think about, this is a slide that or images that I pulled out of spatial sequencing. So it's looking at gene expression in tissue, but I think this is actually quite informative when you think about these proteins. It's like, where in the skin are they found? This is the baseline, but here you see in orange or pink expression of the JAK3, here in purple JAK1, and then here in red the JAK2, and then I have a little bit of a bigger image here for TYK2 shown in yellow. So each dot represents the where the expression is found and where in the skin is actually happening. And the first impression that you get when you see an image like this is that it's actually pretty widely expressed, and it's both in the epidermis and also on the inflammatory cells. This is psoriasis. So you can really appreciate that this is a signaling machinery that is active and operating across multiple different cell types, both immune and also skin cells, that mediate and respond to the cytokine and inflammatory signal in the tissue. So kind of going back a little bit to the basics, so the JAK-STAT signal transduction pathway. So this is kind of how it works. You have an inflammatory cytokine here. And when the cytokine approaches the cytokine receptors, you have polymerization of the receptor molecules. Then you have activation of the kinases that they bind to the inside of the cell, the intracellular region of the receptor, and then they cross-phosphorylate each other. And then what happens next is that these then in turn phosphorylate the receptor that allows the STAT proteins to really approach and bind to the receptor. And then they in turn kind of get phosphorylated. And when the STATs get phosphorylated, they can form these dimers and then kind of move from the cytoplasm into the nucleus of the cell, where they can activate genes transcription program that then mediates the inflammatory signal from the cytokine outside the cell. And if you look at the JAK-STAT family as a whole, you can really appreciate how broad it actually is active in the cells. We're looking at different combination of the JAK. These always pair up on the inside of the cell on the cytoplasmic part of the receptor, and then mediate different signaling from different cytokines. And it's a very broad range of both cytokines and growth factors that actually signal through this machinery. And that involves T cell and B cell function, airway reactivity and mucus secretion in response to helminths. You have differentiation signals that have a role in both myeloid T cell and lymphocyte, hematopoiesis, so generation of red blood cells, platelets, neutrophils, lymphocytes, even growth and metabolism is also involved in the JAK-STAT signaling. Acute phase responses, effector function, metabolism, T cell differentiation, even some degree of anti-inflammatory response, barrier function of the skin, B cell activation, function of cells in the joints and in the bones, antiviral responses, and then effector function of both T cells, lymphocytes, and macrophages. So a very broad category of different inflammatory and immune-mediated signals that really get activated by a whole range of different cytokines and growth factors, but all go through this JAK-STAT type signaling. So what about TYK2? TYK2 is more kind of a narrow, so it's just one of those four JAKs, but it has a key role in some of the key inflammatory pathway that we know are critical to multiple different immune diseases. So TYK2 here binds with JAK1 and mediates interferon type 1 interferon signaling. It's also very active in terms of signaling downstream of IL-12 and IL-23. This is what really drives IL-17 responses that we know are critical for psoriasis pathogenesis. IL-10 and IL-20 family of cytokines goes through TYK2. Some of the cytokines such as IL-6 and IL-11 and IL-22 also go through TYK2. And then they're also involved in signaling and responses to what we call Th2 cytokines that have been implicated in diseases such as atopic dermatitis. And if you look at this kind of a side by side, I find this slide to kind of be, in a way, quite helpful. So if you try to kind of separate the JAK1, 2, and 3 pathways from the TYK pathways, you can see the JAK1, 2, and 3 pathways are involved in both immune responses, but also responses to various growth factors and hormones. So it is both immune function and then broad systemic kind of role that the JAK1 and 3 actually have. Whereas if you look at the TYK2 pathway, it's more restricted towards immune responses and immune function, particularly, like I mentioned before, the type 1 interferon, IL-12 and IL-23 signaling mechanisms. What also is quite helpful is to kind of when we are looking at these kind of pathways, is to kind of look at what pathways are shared between different diseases. And this is obviously a very much simplified kind of a slide, and it's getting a little bit old. It's about 12 years old. But you can see here, if you look at the IL-23 pathway or TNF or type 1 interferons, there's a whole range of different diseases that kind of share and utilize this pathway in their pathogenesis. So IL-23 is critical for psoriasis, inflammatory bowel disease, ankylosing spondylitis, and there's some evidence of it in being involved in rheumatoid arthritis, type 1 diabetes, lupus, and even multiple sclerosis. And TNF, again, cuts across multiple different inflammatory diseases, and same with type 1 interferon, particularly autoimmune diseases such as lupus and multiple sclerosis. And here, what you have on the right is kind of looking at it in terms of the genetics and where there's an overlap between predisposing genes to these diseases. Or when you can see here is the proportion of genes that overlap between the different diseases, where they're positively kind of work in the same direction. It's shown here in green, and when it works in opposite direction, it's shown in orange. But you can see there's much more kind of a green across psoriasis against diseases such as RA, type 1 diabetes, inflammatory bowel disease, and celiac disease. So in terms of the biology, both in terms of the cytokines that I have here on the left, and in terms of the genetic pathways that have been implicated and involved, there's a huge overlap. And this is one of the reasons why many of the drugs that we use to treat one disease will often work in other, related immune and inflammatory diseases. So this is a study that was published in Nature Genetics. This is probably about 10 years old. Shows you some of the new kind of a genetic loci, and I want to highlight here the TYK2, which is on chromosome 19. Actually quite significantly associated with psoriasis. And this is a Manhattan plot. This is actually quite common way to kind of visualize genetic predisposition. And here we're looking at the significance on the p-value, and here we're looking at the different chromosomes. And you can see these different peaks. That's why they are called like a Manhattan. It's like the skyline across New York. And you can see the different peaks that kind of meet certain threshold, and the TYK2 is right here on 19p13 as shown over here. And if we look at the different mutations that have been implicated in TYK2 and associated with psoriasis, almost all these mutations are found in these two domains of TYK2, the kinase domain over here and the pseudokinase domain over here. And these psoriasis variants, instead of predisposing, they're actually protective. So having mutations in these domains of TYK2 will actually protect against development of psoriasis. And I think that's an important thing to kind of state, and I'll be showing you a little bit more data on that in a few minutes. And if you think about where this kind of fits in the pathogenesis of psoriasis, this is a very complex slide showing you how predisposing factors, disease initiation, and disease maintenance kind of plays a role in kind of activating and sustaining inflammation in psoriasis skin. And if you look at the different genes and the different pathways and the mechanisms that are involved, whether it's interferon antiviral signaling, epidermal differentiation, autoinflammatory responses, Th17 differentiation or IL-17 responses, antigen oxidative responses, TNF, IL-23 or T cell development, you can really see that sometimes TYK2, what I'm highlighting here, can be involved in the same disease process, but in multiple different locations. And that often can kind of indicate that a drug would be then more likely to be effective and successful to treat that particular disease. And this is kind of going a little bit kind of a deeper into the genetics of TYK2 and these autoimmune or different autoimmune diseases. So there are multiple different genetic signals that are found in the TYK2. I'm showing you here like three of them in the orange, the blue, and the green. The two main psoriasis one is this P1104A and I64AS. And these are kind of identified with what we call like a fine mapping alleles. And if you look at the contribution of these genetic variants on the risk, the odds ratios of developing those diseases, what you can see here is the one that is 1. So anything above here is predisposing. Anything below in this direction actually is protective. You can see for patients that have one copy of any of these variants, they have lower risk of multiple different inflammatory diseases, including psoriasis, ankylosing spondylitis, celiac disease, or multiple sclerosis. And if they have 2 copies, they have much higher level of protection. And then the effect is much less with some of the other variants, and sometimes a little bit of a mixed one. And you can look at this across the different diseases. So here I'm showing you these 3 genetic variants in the TYK2 that have been shown to be protective or associated with these different autoimmune diseases. And here you have the different diseases. So here, more in blue, that means there's a protection of against developing that disease. So these are protective variants. And then the ones that are shown in red increase the risk. So you can really appreciate the complexity by just looking at these 3 different genetic variants in the TYK2 gene and how differently they contribute to the risk. So you can see this particular variant protects against a whole range of different autoimmune diseases, including psoriasis, rheumatoid arthritis, lupus, and a whole range of other ones. And then you have one that is protective against some diseases, but may potentially increase the risk of other ones. So it's a very delicate kind of a balance and complex and still incompletely understood, actually, how the different mechanism is. But it's very interesting to kind of look at it in this kind of a context, looking at it, the risk of autoimmune disease and then the risk of infection. Whenever you are targeting something that is immune related, you always worry about, if you knock it or suppress it too much, what is the risk of actually getting like an infectious disease? And this is a paper that was published in Science Translational Medicine in 2016. So if you have the wild type allele that is shown here as TYK2++, you actually have a high risk of autoimmune disease. If you have any of these heterozygous mutations, particularly one in the pseudokinase domain, you have much lower risk than if you have a complete absence. So complete absence here of TYK2 will increase risk of infection. Having too much of it will increase the risk of autoimmune diseases. But having any of these variants over here, you kind of get the perfect balance between the two, where you are protected against developing an autoimmune disease, but the suppression is not that high that you're going to be at a risk of developing an infection or infectious disease. So that kind of goes back to, how do we target TYK2 therapeutically? So I want to highlight the allosteric inhibitors of TYK2. So you have these 2 domains of the TYK2. You have the pseudokinase domain, and then you have the kinase domain. The kinase domain is the one that acts with this ATP. And this is called the catalytic side of TYK2. And this is where TYK2 inhibitors that target the active or catalytic domain, that's where they act. But then you have these allosteric inhibitors that actually target specifically the pseudokinase domain. And what the pseudokinase domain inhibitors or allosteric inhibitors do, they bind to TYK2 on the pseudokinase domain. It changes the conformation of the overall protein, the overall structure of TYK2, so it can't really block ATP. It can't really bind ATP anymore. So it blocks its binding and thereby inhibits TYK2 activation. And this is and the reason why it's kind of important to kind of distinguish between those 2 mechanisms is if you look at the active domain of TYK2 against JAK1, JAK2, or JAK3, is actually highly similar across different family members. If you look at the regulatory or the pseudokinase domain, it's very different across family members of the 4 members of the JAK family. So that means that whatever targets the pseudokinase domain is much more specific than anything that's trying to block it in the active domain of TYK2. And the reason for, and the implications of that are like I show over here. These are different tyrosine kinase inhibitors. But what you will see is this is the family tree or structure of the tyrosine kinase family. And you will see that many of them they bind their target, but they also bind a lot of other tyrosine kinases. So they're non-specific. And that kind of goes across multiple different ones that, yes, you may have one major domain binding, but again, it kind of spills over into multiple other ones. It was very, very difficult to kind of get that specificity in terms of targeting or blocking this pathway by inhibiting the active domain. And this is one of the reasons why many of these may have more substantial side effects because of this non-specific binding. And this is kind of shown over here, different JAK inhibitors that we are currently using in the clinic. And you can see that they're binding to different JAK family members and even TYK2 with different degree of affinity. So you have ones that are more JAK1, and then you have other ones that are more kind of a JAK3. So you kind of get a different balance between which JAK they're preferentially targeting. and some hit multiple of the JAKs, and that leads to broader suppression of immune responses and therefore higher risk of potential side effects and complications. And this kind of shows you pretty much the same thing, the non-specific JAKs, but I want to highlight here an allosteric inhibitor over here. They're much more specific to just TYK2 and really do not bind any of the JAKs. So you get a much kind of a cleaner inhibition of the target molecule with the allosteric inhibitors than the ones that are actually targeting the active domain, the tyrosine kinase domain of TYK2. So kind of going back to the immunopathogenesis of psoriasis, it's a complex slide, but I find this to be kind of quite helpful to have an idea where TYK2 is operating in the disease. And I think psoriasis is a prime example because that's the best and the most studied disease where we know TYK is active in. So here you have plasmacytoid dendritic cells that are actually activated to type 1 to different RNA protein complexes to kind of activate type 1 interferon signaling that in turn activates myeloid dendritic cell, and you get Th17 activation or Th1 activation, and then you get these various cytokines that act on the epidermis to kind of then amplify and sustain inflammation in psoriasis. And if you look at the different blue boxes that I have here, these are the ones that have been shown to predispose to psoriasis, but kind of aligned along the different biological mechanisms in psoriasis. So you can see that the TYK2, it's over here involved in IL-23 signaling, therefore blocking IL-17. It's also on the IL-12 arm and affecting both TNF and interferon gamma responses. And then it also inhibits type 1 interferons. So again, you have a way for the same pathway and same molecule to kind of act on multiple different levels in the same disease. And this kind of shows you pretty much the same thing. You have type 1 interferons over here. It binds the receptor, kind of goes to the TYK2 and JAK1, activates STAT1 signaling. This in turn will lead to IL-23 production that will act on Th17 cells and then drive IL-17 production. And then you would get excessive proliferation and activation of keratinocytes that then sustain the inflammatory activity in psoriasis. And the same thing is also happening in the joints. So it doesn't have to be just in one site in the body. It can also be in synovium where you have type 1 interferon acting the same way. Macrophages all contributing to IL-23. IL-23 also goes to the TYK2, activating the STAT pathway, and then through secretion of IL-17 will lead to activation of the synoviocytes. You will get the involvement of additional and other inflammatory cytokines, including TNF. This will activate osteoclast and will get the bone resorption and the bone damage that you can see in this disease in the joints. Again, this is from one of the earlier kind of clinical trials and kind of the molecular response of TYK2. This is looking at different doses of deucravacitinib, which is one of the allosteric inhibitors that target the pseudokinase domain. This is looking at the percentage change in PASI from baseline. So you can see that they're actually quite effective, and you can see a very nice kind of a dose response compared to placebo shown over here in gray. And they're actually fairly fast acting. Within a couple of months, you're actually getting quite substantial clinical response and clearing of psoriasis. And if you look at what are the signals that are really being turned off with this kind of a treatment when you target TYK2, this is kind of looking at different modules or different inflammatory modules. So it's a composite of multiple different genes. But what you can see over here is that with treatment, you get a very rapid response. Of course, multiple different IL-17 modules. For IL-17, you're suppressing IL-22, which is the IL-20 family. And then you're also getting suppression of type 1 interferons. So you're suppressing, with this kind of a treatment, multiple different inflammatory pathways. And you can see this also kind of reflected in terms of the clinical response in psoriasis. So here you're looking at placebo. Here you're looking at deucravacitinib, the TYK2 inhibitor over here. You're looking at non-lesional, the lesional baseline, 15 days after treatment, and then 85 days after initiation of treatment. And you can see the placebo is staying pretty much the same. Very dramatic response in terms of the psoriasis, both in terms of the histology, cell proliferation, or keratinocyte proliferation, infiltration of T cells, dendritic cell, and cell proliferation. So kind of going back, what about the clinical trials that are kind of ongoing? What this table shows over here are the current JAK and TYK2 agents that are currently approved for treatment of inflammatory diseases. So there's quite a few here for alopecia areata and also for atopic dermatitis. But the one that is currently of the TYK2 inhibitors is the deucravacitinib that is approved for treatment of psoriasis in adults. And that's the one that is currently has an ongoing phase 3 clinical trials in pediatric psoriasis in patients with moderate to severe psoriasis in 4 to 17-year-olds. And if you look at the other diseases that are currently undergoing clinical trials using TYK2 inhibitors, there's actually multiple ones. Unfortunately, all of these are in adults. But for deucravacitinib and allosteric inhibitors, there's studies ongoing in alopecia areata, Crohn's disease, lupus, and cutaneous lupus. Brepocitinib, which is more of a mixed TYK2 JAK1 inhibitor, so it acts on the active domain of the TYK2. That's being looked at in lupus, uveitis, dermatomyositis, and scarring alopecias. And then we have some newer agents that are now both allosteric inhibitors that are being or undergoing phase 3 clinical studies in psoriasis and psoriatic arthritis, and then one that is specifically designed to kind of treat CNS disease such as multiple sclerosis and is also in very early clinical trials. So to kind of briefly summarize, so TYK2 is an important on and off switch for specific cytokine signals, the key ones being IL-12, IL-23, and type 1 interferons. TYK2 has a genetic link to a whole range of autoimmune diseases and really helps to kind of establish it as a key pathogenic circuit in this disease. And there's several clinical trials ongoing for a range of different inflammatory diseases. Only one still on pediatric psoriasis, with the other ones mostly focused on adult population for now. But overall, and this is kind of speaking to the clinical experience with the TYK2 inhibitors, particularly the ones that have been on the market for a few years, and this applies particularly to the allosteric inhibitors, is that the side effect profile is very benign. These drugs have established very good long-term safety, and I think we're looking at these being incorporated more and more into pediatric practice, and I think that's going to happen in the near term as soon as those clinical trials get published. So that concludes my talk, and I'm happy to discuss and take questions. So thank you.
Thank you, Dr. Gudjonsson. That was really a outstanding overview of the basic science, the complexity of the genetics and the immunology, and then possibilities for clinical use, particularly in kids, in addition to adults of some of these therapies. While we're waiting for some questions, I certainly have a couple of questions that have come to mind as a pediatric dermatologist and a clinician who sees lots of psoriasis patients. It's really exciting to think that we may have additional treatments, particularly treatments that are oral, tablets, which are really important for children, and because of their dislike of injections and so forth. But I want to take a step back to ask you, you showed us a really fascinating slide and discussed that certain TYK2 mutations can be protective against psoriasis. And my understanding of the literature is that there are multiple, over 500 known modifications in the TYK2 gene, and some might be protective and some might be risk factors. And do we know, or do you know if we understand the basis for when a certain mutation might be protective against in the context of a complex human and all of the other genetic and environmental factors and so forth?
Yeah, no, it's a great question. I think the commonality between the mutation that we have looked at in psoriasis is that what they have in common, the protective ones, is that they decrease the activity of TYK2. So I think it's like the genetics are kind of modeling what in a way getting with the therapeutics. And at the same time, they're not really shutting it down completely. So the protein is just not functioning quite as well as before. So just a drop in the activity seems to be the key. What complicates things when you're looking at the genetics is that many of these different genetic variants are also in what we call like a linkage disequilibrium. So they're all connected. So when you inherit one, you inherit the other one. And therefore kind of sorting out what one, each one of them actually do can be quite complicated, at least kind of functionally. So again, many of those studies have never been done, and I'm not sure when or if and when they're going to be done. But there's certainly, parts where, we know that there are mutations that can sometimes increase the activity of these molecules or increase their binding to the receptors or make them more active in terms of, phosphorylating the stats. And those are probably the ones that are, activating mutations could predispose to the disease.
Right. Yeah, the holy grail is, which we may never achieve, hopefully we will in our lifetimes, would be a form of personalized medicine whereby you can identify specific mutations in a specific patient, have an overall immunologic analysis, and be able to sort out what is active, what is underactive, and what targets to modify.
Yeah, exactly. And they just get enormously complex very, very quickly, and sometimes you can, also, even though this has not been studied yet, some of these mutations may even change the binding of TYK2 to the drug also. So, and
Exactly.
that's another level of complexity that
Right.
adds on.
Yes, that's right. So it's helpful for our audience to know that though linear pathways are shown and mutational analyses are shown, there's such a complexity that the 1x view and the 1000x view are very, very, very different. And I appreciate you helping to untangle and demystify some of that. One of our audience members has a question. I'd like to read it now. Given the unique immunologic landscape of children compared to adults, do you anticipate any differences in the efficacy, safety profile, or infection risk of TYK2 inhibitors in pediatric psoriasis? A second part is, how should this influence our approach to long-term monitoring in younger patients? Thank you for that question.
Yeah, no, that's a great question. And that's always the tricky thing with these drugs is that whenever we initiate them, it's a long-term plan. We know that none of these drugs really cure the disease. They're just very, very good, often at controlling them. I would say, there's at least 4-year, if not 5-year data now available on the one that we have had for the longest, the deucravacitinib. And the safety profile of that actually looks very favorable. But again, it's in adults. So again, take that, with that, I would say that you can't always extrapolate from the adults to the kids. But so far, it's looking very, very promising in terms of long-term, both maintenance of the treatment response and also in terms of the long-term safety of the drug. So I think it's looking very favorable. If we look at some of these mutations that kind of in a way reproduce what we see with the drug, there are patients that live with many of these variants without any health consequences at all. And I think that's always kind of reassuring when the genetics kind of model what you would expect in long-term treatment with the drug. So I think that's an additional kind of thing that kind of goes with the beneficial profile that we have of these drugs so far. In terms of the disease modification, that's something that is, a hot topic at the moment. I think with the data that is available, particularly from the GUIDE study, is that early intervention with an effective drug may change the trajectory of the disease. And I think we still have a lot to learn from that, and particularly in terms of which medications to pick and use to try to change that trajectory. I think the data is just, we don't have it yet, but I think it's being generated. We'll have it probably in a few years' time, and I think that will give us a much better idea how we should approach, new psoriasis diagnosis, which drugs to use, and where can we expect that the disease trajectory is going to be, going to change.
Yes, and I appreciate you. I appreciate you underscoring that we don't yet know how TYK2 variants might impact disease onset or progression of psoriasis, and it would be nice to know that. And in fact, I think as pediatric dermatologists, we have particular concerns because we know that certain mutation in TYK2, as you mentioned, that sits on chromosome 19, can create an immunodeficiency syndrome. An autosomal recessive primary immunodeficiency, and which is characterized by increased susceptibility to mycobacterial and viral infections. And this worries us because of the multiplicity and complexity of this gene and the pathway. And you showed beautiful illustrations of how proximal TYK2 is. And so, in my mind, if you are blocking TYK2 that proximally, you would expect a rather robust downstream impact of regulation or dysregulation. And so, how are we certain, or which, of course, we can never be certain, but knowing that there can be increased risk for viral infections, we know this mutation can cause an immune deficiency. Might there need to be some sort of special screening in advance of using a TYK2 inhibitor in patients so that we don't increase their risk of catastrophic infection?
That's a great point. And again, that's always the concern that if you block something too well, you might have that adverse consequence of in a way inducing immunodeficiency. And I think that's where, those studies like the one that I showed from the Science Translational Medicine, where you have on one end, you have the risk of autoimmune disease, on the other end, you have the risk of infection. And then there is this kind of area or the zone in between. And that's really where you want to hit when you have a drug, that you're not really shutting it down completely, but you're kind of modulating so it's like less effective. And I think the small molecular inhibitors actually are quite good in that sense that you're not getting complete shutting down of the molecule. These are competitive inhibitors of the common kind of a goal. And you get like a partial suppression of the of the protein or TYK2. So you kind of you balance it. But again, obviously, if you go too high with the dose or the drug is just too good, then you might have an increased risk of actually having those types of consequences. So that's why continued monitoring of potential side effects is so critical, even post, particularly post marketing. And I think, and one other thing that I wanted to kind of add is also the good thing with the orals is that when you need to kind of they get out of your system pretty quickly also. So you can reverse the situation much more quickly than you can do with a biologic, for example.
And I think that's a really important point because, the oldies but goodies, the drugs such as methotrexate, cyclosporin, and acitretin, though they've fallen into somewhat less use, the advantage of an oral medication is the turn on, turn off. And I think that you make a really important point, and that's really important in children as well. Dose modification, the ability to stop the effect and not have something in the system for months and months. And so I appreciate those comments. We have a couple of questions from our audience members. I think this is a really practical question that I've thought of myself as well. One of our members asks, why is it called TYK2 and not TYP1 or JAK4, for example? We know that these molecules sit in close approximation and work with the JAK kinases. So any idea why this is named TYK2? Is there
the quirks of protein and gene nomenclature? It probably was, that was the earliest one that was identified. That's one thing. And I think it was a different group than the one that actually identified JAK1, 2, and 3. So it's just a happenstance. Why exactly it was TYK2 and I can't remember that story, but okay. Different labs, different timing. And then people sometimes think these things whatever they want, and then it sticks.
Mm-hmm. Yes, I want to offer a general point and then ask a question from one of our participants, which is that I really appreciate the fact that there are studies in pediatric patients. Not that long ago, pediatric patients were not looked at in clinical trials. There had to be years of adult data post-marketing before a pediatric patient was even enrolled in a clinical trial. And I just want to applaud industry and researchers for having the recognition that kids are not just little adults, that we cannot continue to borrow data from adults and apply it to kids because it doesn't work that way. Kids, in fact, have a very different pharmacology, pharmacodynamics, and handling of certain medications. And so I appreciate that you're showing the data and talking about that and that we have kids in trials. One of our participants wants to talk a little bit about, for example, apremilast, a PDE4 inhibitor, which is another oral agent relatively recently approved for pediatric psoriasis. We know that and I'm going to paraphrase your question a bit, we know that in adults, deucravacitinib is compared with apremilast. Do we know of any comparison yet with pediatric patients regarding the differences in efficacy, safety, tolerability? And that is to say, in what clinical scenarios do we think we might favor a drug like a TYK2 inhibitor in a pediatric patient versus some of the other medications like apremilast?
Yeah, no, that's a good question. Well, I think, overall, if you look at the side effect profile, and I'm kind of referring more to the adult population because I'm more familiar with that. But the side effects, like with the depression, the GI side effects, the diarrhea that we see with apremilast, that's not a feature of TYK2 inhibition. So in many ways, it's probably it's an easier drug to kind of tolerate. But more importantly, I would say that it's also a lot more effective. TYK2, particularly deucravacitinib, is kind of has a similar level of efficacy as some of the like the older biologics that we used to use, like the etanercept, and even up to like levels obviously with Humira. So I'd say that you get a better response with TYK2 inhibitors than you get with apremilast. But that being said, it's very hard to say, which, when should you use one and when should you use the other one. I think that's a tricky one. I've struggled with apremilast myself. I've used apremilast to treat, used it to treat patients that have more severe involvement of their hands and feet. And I've had, good success with that. Sometimes kids or patients that have more scalp involvement, I've had really good response there. But I think overall, my impression with TYK2 inhibition, particularly deucravacitinib, is that it's an overall more effective and better tolerated drug. But again, it's probably more immunosuppressive at the same time, whereas apremilast is more immunomodulatory.
Fair. Yeah, I appreciate that response, and I think it also speaks to what we also know as physicians who care for primarily pediatric patients, that pediatric patients that is comprised of probably 4 or 5 different subpopulations. We've got infants, young children, middle-aged children, preteens, peripubertal, adolescents, young adults. And so we will have to determine, based on a growing, developing child, a developing immune system, where this drug may fall into clinical utilization. Because I must confess that the proximal blocking, and as you said, it's not perfect blocking, otherwise these drugs would be too dangerous to use. But looking at the proximal impact of reducing regulatory molecules beyond, including almost all of the cytokine pathways that are involved in the pathogenesis of psoriasis, gives one pause to think these could be the best molecules ever, but also can carry certain risks. And I want to share and underscore that I think that will be really important going forward, particularly looking at trial data stratified by age subpopulation, because I think you had mentioned that this drug, one of the drugs is in trials for kids 4 to 18. And I think those subgroup analyses are something that pediatric dermatologists look at and consider incredibly important. And I don't know if you have a comment on that at all.
No, I absolutely agree. And I think it's, you have to be careful with kids. And also, just speaking at it from like the genetic, side, when we have looked at like the genetic contributions, the kids that get psoriasis early, they have a little bit of a different genetics than the adults that get it later in life. And they seem to have a higher, what we can call it like a genetic burden. They have higher kind of a carriage of those particular susceptibility genes. And that also will lead to the disease to be a little bit different. And that's an aspect of psoriasis that is just not well explored. We really haven't done much on it beyond just the genetics. But for sure, it's going to affect how they respond to treatment and also the long time, the disease trajectory and how it's going to behave.
Right. That's right. We're getting a little close to time, and I wonder if our audience members have any other questions, or Dr. Gudjonsson, do you have any final words or summary statements that you'd like to make before we close this webinar?
No, it's been very enjoyable. I really, enjoyed the interactions and discussion. And I think this is always a moving field. There are more TYK2 drugs coming, both mixed TYK2 JAK inhibitors and then also more allosteric inhibitors. And some are more effective than others and different. So I think this is going to be something that's going to be very dynamic going forward. And obviously the pediatric studies are just starting. So I think we will have a lot more to discuss just a few years down the road.
Well, and I when I think about the start of my career, and even we're focusing a lot on psoriasis here. And of course there are other diseases that we could have talked about. We both practice complex medical dermatology, mostly in adults for you, and mostly in kids for me. But there's a lot of promise of these drugs, and we're seeing use of these drugs, as you had mentioned in your talk, and in other populations. And I think a closing comment I'd like to make is that, I appreciate that we covered the complexity of these diseases. And truly, we have limited knowledge of the pathogenic processes and the diverse genetic predisposition of patients. And so it's hard to assign causality of a disease to a single variant of a single patient, and the role of triggers and environment and diet and all of the things. There's so much. And I think that's what makes these types of conditions really exciting to think about, where there's room for innovation, and we certainly need newer, better, and safer therapeutics. And so I can't thank you enough for bringing this information to us, this cutting edge research and the knowledge of TYK2 and its role in inflammatory skin disease. So Dr. Gudjonsson, my deepest appreciation to you, and I will turn it back over to Jen Dawson from PeDRA. Thank you.
Thank you.
Thank you so much to Dr. Kelly Cordoro for moderating this webinar, and a special thanks to Dr. Johann Gudjonsson for an excellent presentation. This is an independent medical education program. PeDRA is solely responsible for the content and the selection of all moderators and speakers. Thank you to Bristol Myers Squibb for supporting this program through an independent medical education grant. If you have questions or comments, please email us at info@pedraresearch.org, and make sure you're following us on social media at PeDRA Research so you never miss any new content. Thank you so much for watching.